Hello, I'm Robert Walker, and this is a presentation for my paper about why legal processes will require Mars samples to be sterilized if they return before 2040, or else they'll need to be isolated from our biosphere and studied telerobotically, and I recommend a ring plane above GEO. I'll put a link to the paper in this presentation in the video description, and I'll start with a summary graphic. So I've written out in text pretty much what I'm going to say. It won't necessarily be word for word. So if the process starts in 2021, then the earliest legal return will be 2033, I reckon. But then if the bill has to start after the legal process, it'll be 2040. They want to return by 2031, so the Perseverance rover is already on the way to Mars, and then ESA is going to be sending its ESA Fetch rover and the Mars Ascent vehicle in 2026, along with the Earth Return Orbiter, and they want to return unsterilized samples 2031. That is the sample receiving laboratory, and the facility will be ready for take at least 11 years, legal process at least 8 or 9 years. However, the technology doesn't yet exist to build the facility, I discovered. It must contain starvation-limited bacteria of less than 0.1 microns. And NASA only knows what to build after they complete the legal process. So this, in a nutshell, is the entire paper and what it's about. And so I'm going to embed short videos in the actual presentation itself, and that, I think, I'll just outline a little bit more, then I'll upload this. I think I'll keep this first one particularly short. So this is the ESF's, they say that a particle of 0.05 microns can't be returned, can't be allowed to escape from the facility under any circumstances. Here's the actual words. Release of a single unsterilized particle larger than 0.05 microns is not acceptable under any circumstances. And the reason is because this discovery of this bacterium passed through, this is a 0.1 micron nanopore, and this bacterium passed through it. This is starvation limited, so most bacteria would be too fat to go through a nanopore like that. But Martian life will of course be starvation limited, and so it's quite feasible that there could be a microbe from Mars that can go through a nanopore like that. And that's why they say that 0.05 microns is not acceptable under any circumstances. Now, I looked at the best filters for 0.05 microns, and this is one designed for SARS-CoV-2, the COVID-19 virus, and the smallest one can be 60 nanometers across, as you can see there. And this filter can filter out 88% of particles of that size. That is nowhere near the requirement that we need. And this is an experimental filter in the laboratory. The actual HEPA and ULPA filters are not even tested to this size range, and they're nowhere near this capability. They work by filtering out the larger droplets and then reducing the viral load, because our body, our innate immune system, can fight off a single virus. So the ESF study, their technology doesn't exist yet. They also discovered, in their review of the science, and they found that gene transfer agents of 0.03 microns can transfer overnight, capability for antibiotic resistance to unrelated bacteria in seawater. And they say that there's need for periodic review. And this terrestrial life, they worked out that that's about the minimum size possible for a terrestrial microbe with our biology. But our biology is not nearly, early life can't possibly have used modern biology. The DNA needs enzymes to transcribe it to get the messenger RNA, and then those need the ribosomes to make the proteins and enzymes. Ribosomes also need enzymes and ribosomes to transcribe DNA to make ribosomal RNA, and then their own proteins and subunits also have to be made in the same way. With so much interdependence, it simply can't arise from non-living chemicals in one go. It's got to arise from some simpler form of life. if you look at the size of that simple form of life, then you find they can be as small as 0.14 microns, or 14 nanometers in diameter. And so that goes back to 1996, but there's been more research since then to make that understanding more robust. And then, so the minimum requirement may well be reduced to 0.01 microns, turning all of this red all the way down to here. It's all unacceptable down to there. So we need to do this review first and find what is the minimum size. Then we have to design the technology, which doesn't exist yet, and 0.05 microns is a challenge, and 0.01 microns would be even more of a challenge. This is well below the optical resolution, which is 0.2 microns, of the best diffraction limited optical microscopes. And NASA can't start the build, naturally enough, until they know what they're going to build. For something that costs more than $1 billion, and this, the best estimate so far, we're getting up to $500 million, and it may well cost more than that. $500 million, not $500,000, $500 million. But it's not only that. NASA are the proponents, so that at least would add several years to the 11 years, or estimate that it already is, and that would take you to the mid-2030s. But NASA can't start, but NASA are the proponent in the legal process. So having gone through all that, done the review, produced the technology, and then they need to have at least a particular idea of the technology before they can really make a good start on the legal process. Then they have to convince, they're the proponent, and they have to convince the concerned parties that what they propose to build is safe and will indeed contain the Martian sample. And that can easily lead to changes in the requirements. So NASA can't assume from the outset that their proposal will pass legal review. And so NASA can't start the build until the end of the legal review, which could take the timeline to 2040 at the earliest. But there's likely to be the 8 to 9 years for the legal review is very optimistic. That's, as partners, ESA would also have a separate process. I can't find any estimate for that timeline. And that would involve the legal systems of the EU, the UK and Canada. And then there would be many international treaties involved, and many other organisations would declare an interest. And NASA have simply never gone through anything like this for any of their space missions. So both of those are likely to be underestimated by several years, so I put 2040 in the title, but it'd be no surprise if it took until 2050. And then I was looking at some more things, the legal review or the next sample return study. So if the sample return study don't look into these things that I mentioned, for sure the legal review will. So it's better to come up with it first before you start the legal process. So I'm going to mention some things that have not been mentioned in previous legal reviews. Sample return studies I think should be mentioned, should be investigated, because I'm sure they're going to come up in the legal review. And so, so far, they're focused on the possibility of release of opportunistic pathogens of humans as the main focus. And that's not impossible. The Legionnaire's disease is an example which uses the same method to invade human lungs that it uses to invade a biofilm. So a Martian pathogen of a Martian biofilm could use the same methods to invade human lungs. Another thing which I don't mention there, but you could get a Martian fungi, and there are opportunistic pathogenic Martian terrestrial fungi. I covered this in my paper, but I haven't mentioned there, and these cause quite a few fatalities amongst vulnerable people. And because they can cross over into the brain if there isn't a good enough immune system. And Joshua Laderberg suggested that our immune system might not recognize extraterrestrial biology. And I look into research into nanoplastics, but they find that a 10 micron, I say actually it's microplastics, I should change all that to microplastics, that a 10 micron microplastic has access to all human organs. So our bodies, because there's no immune system, it doesn't recognize, antibiotics do nothing. the immune system won't notice it until it starts to develop a halo of blood plasma, which could, if there was lots of it, lead to blood clots. So Martian microbes, if you were on Mars as an astronaut, and then your habitat, and there happened to be lots of microbes, because you start to find your habitat a nice place to be, your body would be essentially permeable. It would just go straight through your skin, and straight through your lungs, and straight through the barriers to your brain, barriers to your liver and everything. That is, if our immune system doesn't recognise it, and if it is, our body is as permeable as it is to nanoplastics. The technicians, so that's not mentioned in many of you will be experts in different areas, so do have a little bit of think about that. I have a section about that. My paper, it's just a preprint, it hasn't gone through peer review. So I'm very interested to know, well, that argument about nanoplastics seems to be a new one, and it seems highly relevant because it's a good example of something that is organic in a way, but our immune system just doesn't respond to it at all. And is this correct? Is that a good way of understanding Joshua Laderberg's remarks, that it could be like nanoplastics? And so, as happened with Apollo sampling handling, then the 11 technicians had to be, it's a bit of coincidence, Apollo 11 and 11 technicians, but it was actually Apollo 12 that happened for. They had to be quarantined after a couple was found in one of the sample handling gloves. So I look at quarantine, and Carl Sagan, he made a remark about that originally, that the incubation period for leprosy is measured in decades. And remember I think I said, did I say that with the Apollo 11, then there was essentially no legal process. Not that 89 years, but it was zero. It wasn't even a day. They published the guidelines on the date of the launch of Apollo 11. They were much simpler times than now, and they just did it on the very weak Outer Space Treaty. And so they didn't have any peer review, so there's nobody who kind of challenged them and said, well look, how did you come up with your 14 days for the quarantine period for Apollo? And Carl Sagan said, what about leprosy? Its incubation period is measured in decades. And they wouldn't have had an answer to that. And then also, if one of the Apollo astronauts or technicians became seriously ill, their plan was to remove them from quarantine and take them to hospital straight away. That's actually there. You can see it written down black and white. And it's hard to see how they could do anything ethically do anything else. Because even if they knew that the illness was caused by sample contamination, how could you leave someone to die in quarantine unless you knew for sure that removing them from quarantine would definitely harm other people? But then quarantine does very little to protect Earth. And it's hard to see how any form of quarantine could work, either for astronauts or for technicians. And Typhoid Mary shows how it's possible to have a lifelong symptomless super spreader. So lack of symptoms doesn't mean that the technician is safe to release from quarantine. Even if they're completely symptomless for however long your quarantine was, even if it was lifelong quarantine and then they went out, if they're like Typhoid Mary, they could then be infectious when they leave. And then, as well as that, and this is where I think the World Health Organization, they would immediately go click, 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 that's what they'd be thinking like this. But astrobiologists have a different way of thinking, and nobody seems to have even brought up Typhoid Mary to date. And then, and so I'm just bringing things up that I'm sure would come up in the legal process, but because of the way that people, have specialities, I'm multidisciplinary, and the people who write are always, they're very specialist, the people who've been working on it, and they don't seem to have thought of these things. And then microbes that are harmless to humans, but harmful to other life on Earth, they could become part of the human microbiome. So you could, for instance, have mirror life, and it just becomes part of the human microbiome, but it's harmless. It's very similar to the microbes we have already, and for some reason our mind is, maybe for some reason the human body doesn't mind a little bit of mirror life on the skin. If that was the situation, if it wasn't actually harmful to us, then you couldn't sterilize it. You might even know it's there. You might even sample the microbiome of the technician, or the technician samples his own microbiome, discovers mirror life there, and says, oops, I've got mirror life. How am I going to get out of this quarantine facility? And there would be no way to sterilize a human being of mirror life. And the next sample return study is likely to look into synthetic biology. And that again has advanced hugely in the 8 years since the ESF study. You can already make life that uses Hachimoji DNA. And this has 4 extra synthetic nucleotides, making a total of 8, an 8-base genetic code, which is inheritable and is able to produce an enzyme that causes spinach to fluoresce green. And they've also made the first steps towards construction of a mirror-like synthetic living cell. And that's led to a discussion of the safety requirements of this research, with some synthetic biologists arguing that the 1 in a million requirement of a biosafety laboratory is not sufficient for synthetic life. Instead, they design the organisms so that they require chemicals only available in the laboratory and couldn't survive in the wild. This is not a solution for mirror life returned from Mars accidentally, because we can't change its biology. And the synthetic biologists warn that if synthetic mirror life is able to metabolize terrestrial organics, the result is not pretty. The thing is that with the exception of a few microbes from Antarctica, terrestrial life would not be able to metabolize the mirror organics. So you have the mirror life able to eat terrestrial organics, and the terrestrial organics life can't eat the mirror organics. So everything just goes one way. That the mirror life would turn nearly all our organics to mirror organics, which our life can't eat. And they would make it unavailable to terrestrial life. And that could be devastating for terrestrial biosphere in the worst case. And they outlined that it really is quite dire what they would say would happen. I do believe that humans would survive in that situation. We would basically paraterraform the Earth, covered in glass greenhouses, with habitats inside. And we would find one way or another, we'd find a way to keep mirror life out of our habitats, and or we constantly somehow sterilizing it or something, and then we would be growing the life inside, and then we would be able to preserve nearly all the seeds, because seeds can be preserved in seed banks, and then some of the animals and the birds and things, however many we can manage to save over the number of decades, which could probably take decades and not just years, and maybe even centuries, we don't know, before it's completely devastated most of the terrestrial biospheres. And so we would gradually rescue it. And so I don't think humans would go extinct, but it's not a pretty future. We don't want this to happen. And we don't know why Earth doesn't have mirror life. And Mars could well have both forms, normal and mirror. So I'm not saying it just has mirror, why doesn't it have both? And then most of the organics from the surface of Mars would come from comets, asteroids and interplanetary dust. And those would have organics of both handedness. They sometimes have such like chiral and balance, but they usually have both handedness. So there'd be a great evolutionary advantage for Martian life to be able to eat life, organics of both handedness. So both handedness of Martian life would eat organics of both handedness. So we'd be returning mirror life to Earth, which would be able to eat terrestrial life, but terrestrial life would not be able to eat the mirror life. I think we already talked about these are all the legal process of the World Health Organization. So, but if there's a potential for I already mentioned that, if there's a potential for large-scale impact on the terrestrial biosphere, then the organisation connected with agriculture and fisheries and did almost all aspects of human life might declare an interest. It's hard to think anything that any organisation indeed, that would not declare an interest if there's a potential for destruction of the terrestrial biosphere and replacing it with mirror life. And so that is likely to add to the complexity and duration of the legal process. But then as well as that, the 2012 ESF review considered variations on the precautionary principle. That's the principle that it's the proponent of the project has to prove it safe, even if some of the scientific knowledge needed doesn't exist yet. And there's several variations, including the best available technology, and that's what they used for the European Space Foundation review of the Mars sample return. And they use that together with a 1 million probability of escape for a single particle for the entire lifetime of the facility, together with best available technology to achieve that as best as possible. And if you can't do it, then you do it a serious review and do the best you possibly can. And then they also considered the prohibitive version, where if there is, but actually it turned out the best available technology does not permit their 0.05 microns of unacceptable under any circumstances. So it's a kind of mix between best available technology plus extra requirements to make absolutely sure that with the best available technology you at least achieve this 1 in a million probability of escape is the worst that you permit. And that's basically what the ESF study did. But they also considered the prohibitive version, where if there is a appreciable risk of harm, the sample return is prohibited, unless you can avoid any appreciable risk of harm. And they concluded that this version couldn't be used, as there's no way to prove that there is no appreciable risk of harm. And so this approach, where you basically treat it as like being a biosafety laboratory with something like anthrax or something, you're thinking about something inside that if it does get out, you can actually contain it, and it's probably treatable too, like anthrax, easily treated with antibiotics. So, but Martian life would not necessarily be like that. And this is, I think their approach in the legal process would only likely be acceptable if by harm they mean the possibility of release of an opportunistic pathogen that could harm a few dozen people before it is contained. But Carl Sagan argued that we shouldn't take even a small risk with 1 billion lives, and if harm includes the possibility of a large-scale modification of the entire terrestrial biosphere, then there are likely to be many others, I think, actually, who would take the same view as Carl Sagan. Even people who dearly want to study the unsterilized samples, they have great scientific interest in science, I mean, but still the possibility of even a minute but appreciable risk to Earth's biosphere would just make it impossible. They might highly value science, but they value our biosphere more than they value trying to do science as fast as possible. So the argument in response to this that NASA would put up would likely be the ESF study that we have to use the best available technology because with the prohibitive version the sample return can't be done. But this would be less compelling if there's potential for such large-scale adverse effects. So with this background, I do think it's possible that the facility would simply not be approved at all, even with increased requirements and new technology for the filters. We simply don't have the capabilities yet. So the simplest solution is to sterilize the sample. In one study, using gamma radiation, and it had almost no difference to geology. I'll go into some of the details there. The main thing that happens is that the salt crystals turn blue and quartz turns brown. You can live with that. The details of the geology were the same, and the radioisotope dating wasn't changed at all. And we'd still recognise present-day life. We'd have the carbon signature, we'd have the amino acids, we'd have larger molecules like chlorophyll, and we'd actually see the visual structure. Now, when it comes to past life, again, sterilizing won't do much, because any past life is likely to be degraded beyond the point where it can be recognized. It's not likely that we turn a sample of past life from Mars and the astrobiologists look at it and say, okay, yes, that's past life, we've proved it. Because we already have Martian meteorites with multiple biosignatures, and they actually use the Martian meteorites as an idea of what to look for on Mars. They think like geologists. It's mainly designed by geologists, and geologists think if we can find the provenance of something, that will answer all our questions. It's not the same for astrobiology. No matter, even if you know exactly where it comes from, but you can't prove that it's life, you still can't prove it's life. It's not like geology. And so, many abiotic processes that can mimic the biosignatures in most past life is likely to be degraded beyond recognition. If it's deposited in warm conditions, it would lose its chirality signature, and the organics spontaneously flip to the radiolized form till you've got equal amounts of both. That happens, I think it's over hundreds of millions of years. I've got the figure somewhere. And the certainly billions of years old organics. And if it's warm, yes, like for instance, organics from the time of the dinosaurs to the present day. Now, if it was kept cold in Antarctica, then it would preserve its chirality if it's very cold. But even over that, even tens of millions of years, if we had organics from the ammonites, for instance, and if the Chicxulub impacted sent some of those to the moon, then they might survive there and still have the chirality preserved in the ice at the lunar poles. But apart from that, we've lost the chirality because there hasn't been anywhere that has stayed that cold for that period of time. Antarctica moved to the poles after that. So the chirality wouldn't be recognised if it was warm back then when it deposited, or it came up to the surface and back down again and then up again. And the reactive perchlorates and hydrogen peroxide, they degrade the organics, and cosmic radiation and solar storms. And then you've also got the organic inflow I already mentioned, comets, asteroids, and interplanetary dust, and that is all mixed in with the organics from life. And then the past life is also very likely, actually, to be not have developed photosynthesis yet, and to be limited in its spatial distribution and in time. So it's certainly quite possible that there's only little bits of it here and there, very patchy. So how are you going to find it when you can't detect it in situ, mixed in with all the organics from comets and asteroids and stuff? So astrobiologists, they don't think it's going to find recognisable past life. Even if it's there, they won't recognise it. They won't know for sure that it is. They've designed instruments, many instruments. They could send a suit. Perseverance could have taken an entire suit of a half dozen different astrobiological instruments for the same mass as the MOXIE thing that is making oxygen, which is of no use to astrobiology or to geology. And these would let it to recognise life in situ and then home in and find it and say, oh, this looks like life, this particular rock sample looks a bit like life, let's look around here, see if we can find a more distinctive signature, stuff like that. Perseverance can't do that. So we shouldn't raise the expectations too high. Astrobiologists think of this as more of a technology demo for a future mission of more interest in the discipline. So I suggest using a nanoscale X-ray emitters to sterilize the sample, and those have power consumption of watts instead of kilowatts, and that could lead to a streamlined sample return where the sample container is sterilized during the return mission to Earth, and then it just returned using air capture, using one extra mission. And so I'll stop this at half an hour, and then I'll do another half for this, the rest of this. So if we knew the sample is sterile already, I'll stop it now, and then I'll go on to the second half. And so I'll now talk about how we know that, why we can't assume that Mars is sterile like the Moon, and why what is on Mars, if there's life there, why we don't expect it to have got to Earth either. So I'll do that, and then I'll keep going in the second half of this.